I am very excited to be here to present the results of a series of experiments that we carry out to investigate variation in rat CO2 sensitivity. I think that we all agree that we should kill animals humanely, meaning that while the animals are being killed, they should not experience negative emotional states such as pain, distress, anxiety, or fear, or that at least these emotional experiences are minimized. But how do we know if a killing method is humane? We often use behavior to make inferences about animals' emotional experiences. In this talk, I'm going to focus mainly in three types of behavioral tests that are commonly used to assess the humaneness of CO2, which is the most commonly used killing method for rodents. In this video, we can see a rat being force exposed to CO2. In this type of tests, rats are placed in a closed chamber without the possibility to avoid exposure. The chamber may be prefilled with CO2, or CO2 could be gradually introduced into the chamber. We measure spontaneous behaviors such as increased locomotion, rearing, or pushing and scratching at the lid, commonly known as escape attempts. Here we can see an approach avoidance testing. The rat enters a cage to access sweet rewards, and as soon as the animal starts eating the rewards, a CO2 flow is introduced into the cage. The rat can avoid exposure by escaping to a CO2-free cage, but at the cost of losing valuable sweet rewards. And we measure latency to avoid CO2 and CO2 concentrations avoided as a reflection of aversion. Now we can see an aversion avoidance testing. In this setting, CO2 is gradually delivered into a preferred dark chamber. Rats can avoid CO2 exposure at the cost of being exposed to an aversive bright light. And again, we measure latency to avoid CO2 and CO2 concentrations avoided. So when rats are forced exposed to CO2, there is an increase in locomotion, rearing, escape attempts, and vocalizations. In both approach and aversion avoidance tests, rats avoid CO2 before becoming recumbent or losing consciousness. However, not all studies using forced exposure have found an increase in behavioral responses. In addition to that, behaviors measured in these three settings have shown to be highly variable. For example, during forced exposure in one study, escape attempts range between 0 to 34 between rats. And only 50% of rats increased locomotion in another study. Concentrations avoided in approach avoidance testing have ranged between 11 and 18.6% CO2 among rats. And the latency to avoid CO2 in aversion avoidance has ranged between 7 and 48 seconds between rats. We use behavior to make inferences about rat emotional states. If the behavioral responses when inhaling CO2 are variable, is the emotional experience also variable? We are lucky because CO2 inhalation is very well studied in humans. We know that people experience feelings of fear, anxiety, panic, and feelings of breathlessness when inhaling CO2. We also know that this felt experience is variable. For example, one inhalation of 35% CO2 induces panic in between 12 to 41% of healthy individuals. However, the panic rate is way higher, between 43 to 94% in people diagnosed with panic disorder. Not everyone has the same emotional experience when inhaling CO2, but when individuals respond, this experience is consistent. For example, feelings of anxiety elicited by a prolonged inhalation of 7% CO2 were consistent between two exposures in 60% of healthy volunteers. This is known as variation in CO2 sensitivity. The main aim of this series of studies was to determine if, as in humans, rats vary in CO2 sensitivity. Given the reported variation in responses to CO2 within each one of these three experimental settings or contexts, we first wanted to know if individual differences in responses to CO2 were consistent within each one of these experimental settings. If found, we wanted to know if this variation was consistent across context or experimental settings. We exposed 11 female rats twice to CO2 in each one of these 3 experimental settings, and found that variation in rat behavior was consistent between 2 exposures to CO2 within each one of them. We also found that rats that show a higher response during forced exposure were consistently less tolerant to CO2 in aversion avoidance, but found no evidence for a relationship between aversion to CO2 and approach avoidance and responses in the other 2 experimental settings. In summary, this study shows that the individual rat is a relevant source of variation in aversion to CO2. And from the cross-context consistency, we have some evidence of variation in CO2 sensitivity. But although responses in approach avoidance were consistent between 2 exposures, these were not related to responses in the other 2 experimental settings. Consistency in responses could be reflective of personality traits elicited by the context. So we assess stability and consistency of rat responses to CO2 and approach avoidance across multiple exposures. If we take a closer look to approach avoidance, exposure to CO2 is paired with access to sweet rewards. The rats are highly motivated to approach. We often assume that motivation to approach sweet rewards is similar among rats, but this motivation has shown to consistently vary among them. Next, we wanted to assess if individual differences in aversion to CO2 were related to sweet reward motivation rather than CO2 sensitivity. To assess motivation for sweet rewards, we used this experimental setting. Rats could get rewards hidden in a layer of sand, and we predicted that variation in reward motivation would be related to individual differences in aversion to CO2. For example, rats that are more sweet reward motivated may be tolerating higher CO2 concentrations than those that are less motivated. Let's take a look at approach avoidance contingencies again. Rats are not only provided with access to sweet rewards or gains, but are provided with a safe, CO2-free cage. Evidence has shown that rats consistently vary in their motivation to approach gains, known as promotion, and their motivation to maintain safety, known as prevention. We wanted to assess if the strength of promotion and prevention motivations could be related to individual differences in aversion to CO2. As we can see in this video, in the tests we used to profile the strength of promotion and prevention motivations, rat could approach gains in a treat location or safety in a dark location. We predicted that promotion-focused individuals might tolerate higher CO2 concentrations in order to maximize gains, and prevention-focused individuals might seek non-threatening CO2 concentrations to maintain safety. We started with a pool of 12 female rats, from which 9 reached the training criteria for approach avoidance testing and were exposed to CO2 between 4 and 10 times. Then we assessed reward motivation in 11 rats. 8 subjects were rats previously exposed to CO2, and 3 rats were from the original rat pool. We tested them 3 times in the sweet reward motivation setting. Then we assessed the strength of promotion and prevention motivations in the same 11 rats. We exposed them twice to the experimental setting. In this figure, rat identity is presented in the x-axis, and the average latency to avoid CO2 is presented in the y-axis. As we can see, despite the variation within individuals, differences between individuals are evident. We found that aversion to CO2 is highly repeatable, and that rat identity explained about 40% of the variation in aversion to CO2. We were able to detect consistent individual differences in sweet reward motivation, with some rats consistently spending more time searching for rewards and consuming more rewards than others. We were also able to profile the strength of promotion and prevention motivations. Some rats spent most of their test time in the treat location, while others spent it in the dark location. However, when we looked at the relationship between these personality traits and aversion to CO2, we found no evidence for consistency. In summary, we found that individual differences in aversion to CO2 were consistent across repeated exposures. So, a good candidate for a personality trait. But found no evidence for a relationship between aversion to CO2 and the personality traits that we assessed. So, considering the findings from this study and the previous study, we propose that it's very likely that rats vary in CO2 sensitivity, meaning that the variation in behavioral responses to CO2 likely reflects individual differences in the emotional experience. We know that in humans, this variation is linked to feelings of anxiety, fear, and panic, and that the feelings of anxiety that people experience when inhaling CO2 are decreased by the administration of anxiolytic drugs. We wanted to assess if variation in rat CO2 sensitivity was linked to feelings of anxiety. We took 6 of the rats used in the previous experiments, and we exposed them 3 times to CO2 in approach avoidance. Then we exposed them again, but pretreated with the benzodiazepine midazolam. And as we can see in this image, the drug was administered orally, mixed with vanilla pudding. This figure shows treatment effects and consistency in individual rat responses. Each line corresponds to an individual rat, while the dots represent the average latency to avoid CO2 during control and midazolam treatment. As you can see, the anxiolytic increased tolerance to CO2. In addition, individual differences were still consistent across treatments. In conclusion, we found that it's very likely that rats vary in CO2 sensitivity, with some rats consistently finding CO2 more aversive than others, as seen across all the studies presented here. Aversion at lower CO2 concentrations is likely driven by feelings of anxiety, as shown by the increased CO2 tolerance after pretreatment with a type of drug that in humans ameliorate these feelings when inhaling CO2. Putting these findings in the context of rodent euthanasia, the conjunction of results presented here not only add to the previous literature indicating that CO2 induces negative emotions in rats, but raises other concerns, as between 22 to 33% of the rats in these studies were highly sensitive to CO2. And all rats, even less sensitive rats pre-treated with midazolam, avoided CO2 before losing consciousness. Thank you.